Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another fascinating guest today who is helping to create a better tomorrow. Today we have the honor of being joined by Dr. Andrew Adams, who is currently the Vice President of both neurodegeneration research at Eli Lilly, but also co-director of their new Lilly Institute for Genetic Medicine, which is a fascinating $700 million initiative ultimately to establish an institute for researching and developing genetic medicines, specifically acting at the nucleic acid level to advance this really new drug class that help target the root causes of diseases in approaches that are really fundamentally very different than medicines that are available today. In this role, Dr. Adams is responsible for leading the discovery of various new types of therapies via both internal research programs and external collaborations. And these novel approaches, once again, will allow Lilly to access what are known as previously undruggable targets across a range of the different therapeutic areas that Lilly works in, ultimately opening up new avenues of clinical investigation. In addition to these major roles, Dr. Adams, over the recent years, has taken on significant, substantial leadership roles in both the development of Lilly's COVID-19 neutralizing antibody projects, as well as serving as VP of Lilly Genetic Medicine. And during his time at Lilly, he served roles across discovery, innovation, and really leading these trailblazing teams and championing new ways to bring science to patients with increased speed. Dr. Adams holds his undergraduate degree in biology, his doctorate in zoology from the University of Aberdeen, Scotland. Prior to joining Lilly, Andrew was a postdoc fellow at Harvard Medical School studying things like the neurobiology of anorexia nervosa. Really exciting background and an exciting guest, and we're honored to have him. Dr. Andrew Adams, thank you so much for taking the time to come on the show today.
Thanks, Ira. Looking forward to the conversation.
Yeah, it's as I mentioned already, you do have a fascinating background. I was looking sort of through your publications at some of the early days when you're publishing on things like fibroblast growth factor and hepatic metabolism, various neuroendocrine factors and feeding processes. Obviously, they feed very nicely into the whole leadership of Lilly and adipose biology, energy metabolism, and so forth. Talk a little bit about, if you would, for a couple minutes about the early days. How did you develop some of these interests in STEM and biology? Of course, zoology, fascinating with this focus on neuroscience, and a bit of your sort of, bridge from academia into industry.
Yeah, happy to. I mean, it's kind of an interesting story, and I sort of tell it to a lot of people when I'm mentoring folks here at Lilly, because I think it's kind of a window into how some of the choices you make take you in unexpected directions as you go through your career. For me at the start, I was honestly really passionate about paleontology. That was how I ended up in zoology, because my thought process at the time was that the supervisor that I was going to work with at the University of Aberdeen actually had just published a really fantastic paper on flight in Archaeopteryx. And I was like, that's what I want to do with my life. I want to figure out like paleontology and do things like biomechanics. I signed up for that kind of work with him. But when I came along to start the work, as is typical a lot of the time in scientific labs, the work had moved on and they'd actually switched over to focusing much more on energy metabolism in mammals. And so that was sort of where my research career got its start. Not really where I thought I was going to end up, but once I started doing it, it was just a fascinating way to look at biology, picking apart circuits in the brain to figure out how they work and then putting them back together again to figure out new ways to make medicines for patients was really the first taste I got of this mechanistic understanding of biology. and I was really fortunate then to meet some people while I was doing that PhD in Scotland that led to the move over to Boston and to Harvard, and continued the exploration in different areas of how the brain works and how the brain's sort of central to regulating a lot of these really interesting and important processes.
And, I'm by background training, I was educated as a pharmacist, a practice community pharmacy 30 years ago, but remember sort of Lilly's leadership and sort of the small molecule space, so the Prozacs and the Zyprexas and the Keflexes of the world. Obviously, I look at the top, the leading products of Lilly today, all the larger molecules, the Trulicities and the Taltz and so forth. So, Lilly has been there, like many companies in this space, in this transition from the small molecules to these genetic medicines. And in a sense, you've always been involved in genetic medicines back to the sort of the original days of the humanized insulins and so forth. Talk a little bit, and we'll get into Lilly Institute in a moment, but talk a little bit, as you've been involved in research, been involved in the industry, a little bit of how you've seen this transition. Is it something that, a lot of the people knocking on the doors in business development have brought more antibodies in as opposed to small molecules? Or how did the transition really occur as you saw it being involved in the system?
Yeah, I've been really fortunate to have a 11.5-year career at Lilly at this point. So I've sort of seen a few of these things play out, especially the biologic transformation, right? Like moving from a broadly small molecule based company and small molecule based industry to a much more large molecule focused company at Lilly, as well as with a lot of our peers. And, really, I think the way that this typically works, and you're seeing it play out again now with genetic medicine, is there are usually a lot of things biologically that we really want to do. There are targets that we want to hit. We know sort of fundamentally, scientifically, that if we could do A, B, or C, it would probably be beneficial for patients. But much of the time, you're sort of waiting for the technology to catch up to a point where you can actually do it. That was, small molecules were really good, I think, initially at hitting some of the targets where we had hypotheses around specific ways of manipulating them upwards or downwards. And we went after a lot of those targets here at Lilly and elsewhere. And like you said, that's things like antibiotics, things like vancomycin, or, antipsychotics, like some of the ones that we worked on here at the company. But really, you couldn't do with that small molecule technology insulin or GLP-1 or some of the things that, or Humira, right, for some of the bigger drugs that people would be familiar with. Those are really tough, at least they were really tough historically to do with a small molecule. Then came the biologics, and you sort of had first the explosion in antibodies and a lot of really important therapeutic antibodies being brought to the market. A lot of more recent innovation in peptides as well. Things like GLP-1, I think, are transforming diabetes care around the world. And, we're proud to be part of that too here at Lilly. But the next frontier, the next step, I believe, and I sort of bet on that here at the company with the work we did initially with Dicerna and internally, and now with this new center, is going to be the revolution in nucleic acids and how that's going to expand to new, interesting targets that we couldn't hit even with the biologics.
And continuing along that theme, so we've had the recombinant proteins, we go to the antibody era, we're now the nucleic acids, and obviously we all know about mRNA vaccines and so forth. It's been in the public lingo now for the last couple of years. Talk a little bit, so now we get to the Lilly Institute for Genetic Medicine, a fascinating investment. Introduce us, in essence, what you view as sort of this thing that we call genetic medicine, and then also introduce us to, I mentioned in the sort of the intro, this principle of things that are undruggable, that have been, you were saying, some things are it's not very easy to drug things in the first place, but there's a lot of sort of dark space in the genome that, people scratch their heads, and the chemists, that's hard to drug in the best. Talk a lot about what undruggability means and how some of these genetic medicines allow you to overcome some of those hurdles.
Yeah, that's a really good segue. So maybe we'll go into a definition of genetic medicines as sort of how I think about it myself as a non-genetic medicine expert historically that came into the field and sort of has been thinking about this for a while. So basically what we're talking about when we're talking about genetic medicines are medicines that act on DNA or RNA to affect the result that you get biologically. You can think about this in terms of approved medicines, with things like inclisiran for PCSK9 in cardiometabolic disease, Novartis taking that hopefully to a lot of patients and helping them manage their LDL cholesterol. You can also think about, like you mentioned, the COVID vaccines, right? Like this was really a technological right place, right time as an industry to have this really flexible technology that let us bring forward vaccines in record time. It's probably one of the medicines that's touched the most lives ever at this point, the COVID vaccine tech. So really, in terms of RNA and DNA, it is enabling us to do things that we couldn't do before. And that's this idea of undruggable targets, right? So I'll give you a really good example of something that, we and others have been thinking about here at Lilly. There is a protein heavily involved in Alzheimer's disease that aggregates called tau. And you'll get seeds of tau spreading through people's brains. It's associated with cognitive decline. We believe it's extremely therapeutically important in the development of the symptomatic Alzheimer's disease and the spread of the aggregates through people's brains. The problem with tau, there have been efforts, and we've been part of it, to try and target tau with antibody technology. But the challenge there is a lot of the tau that you want to target is within the cells. So it's sort of hidden from antibodies' reach because they traditionally are not very good at targeting intracellular proteins. And at the same time, small molecules, as you know, it's very tough to make small molecules that penetrate the brain robustly and get to all of the different neurons that we need to get to knock down something like tau. And also to do it in a specific way that doesn't have sort of downsides to it. Small molecules, I think when you hit the right small molecule, it's fantastic. And there are some really great examples of really beautifully designed, elegant small molecules. But making them is certainly a really intense effort that takes a very long time. Tau, though, if there was a way that you could manipulate the genome, all of the transcripts floating around in a cell, and reduce the expression of tau before it's ever made, that would be a really interesting way to target tau from a therapeutic perspective. And one of the technologies that we've invested in heavily here at Lilly and that other people are pursuing as well is siRNA. So it's these short interfering RNAs. And what you do is you take advantage of the machinery that the cell usually uses to fine-tune gene expression. It's this complex called RISC. And the RISC complex basically usually loads microRNAs that your cells make either locally or distally to regulate gene expression, to move up or down the profile of genes that you're expressing at any one time. Usually what microRNAs do is they manipulate several genes at once in a coordinated profile. And so it's like if your liver senses, a hypoxic environment somewhere, or if there is a toxic insult, like you've been drinking alcohol or something, there will be microRNAs that go up and down to regulate the body's response to those situations. And they do it in a really coordinated way across multiple transcripts. The way that siRNA works is, people have the idea of what if instead of regulating multiple genes, you could use the specificity of the genetic code of the sequence to make it so you just knock down the expression of one gene at a time. You load into RISC and basically you use this siRNA as a bait that fishes out all of the transcript that makes the gene that you care about, and then you chop it up and then it never gets expressed. And that's essentially at an extremely reductionist level how siRNA works. We fish out the transcripts, we destroy them through the RISC complex, and then they don't get expressed. So with tau, that's a really fantastic way to think about it, especially because one of the other advantages of siRNA is there is an extreme PK-PD disconnect, so pharmacokinetic to pharmacodynamic disconnect, in that you dose and you have, instead of an insulin or an incretin where you're taking your medicine once a day or once a week, with the siRNAs, because of the way that this RISC complex that chews up the RNA works, it's very stable, and it does that for a long period of time if you load it in the right way. In some of the cases, we're envisaging potentially twice a year you get dosed, and you have a subcutaneous injection, and you go and see your lipidologist, and we'll knock down one of these dyslipidemia-associated genes in your liver, and we'll do it twice a year, and we'll do it in hopefully a way that's convenient for patients, that sort of increases adherence to the drug, because what we're always trying to do is make things more convenient for people to take their medicine and remember to take their medicine. From your background that just having people remember to take the medicine a lot of the time is tough, especially because for a lot of these diseases, we're going into sort of the more elderly population where they have a fairly substantial pill burden already. So when you're adding more things on top of what they're already having to manage as a patient, it can be tough to stay on top of that. So if you can have something where you go to your doctor's office for your regular sort of 6-monthly checkup and you get your shot that helps you manage your lipid levels, and it does it without you thinking about it, right? It's just knocking down that gene for the next 6 months in your liver, or hopefully one day it'll be knocking down the tau in your brain once a year or even less frequently. And it's with that kind of a technology, with both the convenience and the specificity, that we really hope we can go after things like tau that we've known genetically. We really would like to hit that target for a long time. Now we finally have the means to do it.
Fascinating, really fascinating. And as you're talking about, some of these let's say these forms of medicine, of genetic medicines, which you explain them very elegantly, but I'm sure they're extremely complex to how you ultimately put all that together, the risk with the siRNA and so forth. One of the themes that we've gotten into, it's been kind of hot in recent months on the show, is just this whole domain of things like artificial intelligence and machine learning. Obviously, regular drug development's hard. Here are some interesting in silico ways to get things done. Introduce us to some of the tools that are responsible that you can utilize at the Lilly Institute for creating these new genetic medicine forms, because I'm sure it's not something that's easy to put something together. So,
yeah, I think when I talk about it, it definitely makes it sound easier than sort of the hundreds of people we have spending God knows how many hours on actually making these medicines happen. And it's the hard work of all those scientists that really make this possible for patients. And I'm lucky I just get to talk about it with people, all of the work that they've done. So some ideas, some examples I'll give you. The first challenge that you have when it's a challenge, but it's also an advantage, that essentially we're working on a code problem to a degree, right? Like your genetic code is just a series of letters. And so if I need to find something, if I'm going to knock down a gene using siRNA in your liver, say, the first thing that we have to do is we have to pull out the sequence for that gene. And then we have to search through it for like sequences or regions of that gene that will let us effectively target it and knock it down. And so with siRNA, there are specific rules as to what makes a good sequence and what makes a bad sequence, because people have done a lot of experimentation historically. It's the amount of sort of the GC content out of your ATGC that dictates good versus bad in terms of the bases of your genome. There are certain types of repeats that might be bad. You want to avoid things that look like people's own microRNAs, because you don't want to mimic those by accident and then start knocking down things that you don't intend to. And so trying to find these specific yet potent sequences is actually something that gets done with really sophisticated algorithms. And actually it does take advantage of AI/ML approaches to find those right sequences that we predict will be most potent. Because in theory, if you chop up a gene into 21 letter sequences, which is typically the number that you have in one of these siRNA molecules, you're going to get thousands of possible combinations of sequences that you can test. And so really using these AI/ML type approaches, applying the rules that we've sort of built from the literature, but now also refined with the data that we've gotten in previous experiments, we can really, in a smaller number of constructs than would ever really have been possible, predict the potent ones without having to do the in vitro or in vivo experiments a lot of the time these days, which is obviously huge time saving and means we can work on a lot more things. The next piece, I think, of the puzzle that's really interesting to talk about, and it's a focus already in Boston for the team that we have there today, and it will be an even bigger focus as the Institute opens in the coming years, is this idea of nanomedicine. And we've all experienced a couple shots of nanomedicine in our arms at this point with the COVID vaccines. Essentially the idea of using a vehicle made of lipids to deliver the nucleic acid sequence that then gets expressed in the case of the vaccines as antigens for the COVID-19 virus that your immune system recognizes and generates antibodies and T-cell responses to. Right. So in our case, we want to use these little vehicles to take nucleic acid payloads like siRNAs to places that are a bit more challenging than an intramuscular injection for an vaccine approach. In our case, you could imagine that it might be really interesting that instead of today giving you an intrathecal injection for your CNS medicine, and I'll explain that a little bit because people might not be familiar generally with intrathecal injection, because fortunately it's mostly been rare disease that patients have had to deal with this so far in smaller numbers. But basically what you do is very similar to an epidural injection that people will experience as part of pregnancy in some cases. You'll get an injection in sort of your lumbar spine at the lower part of your back of the medicine, and then it will travel up the CSF in your spine and into your brain. That's the best way we have today to deliver genetic medicines to the brain. Yeah. It's cumbersome. It's not scalable particularly easily because you need specialized medical practitioners who are experienced in delivering those kind of injections. And so we really are trying to think about ways that we could deliver those medicines that we need to get to your brain peripherally, and then have them cross the blood-brain barrier, which is this barrier that's specifically designed to keep things out of your brain. So we need to get things across that barrier and then have them, once they get across that barrier, distribute into your brain. We think nanoparticles might be part of the answer to that solution, because they're a way of hiding what is really fragile cargo from the rest of your body, and then taking it to the cell in the brain where it needs to get to and helping it get inside. But even that, it's akin to sort of, I think about it a lot of the time, it's sort of building rocket ship levels of difficulty, right? Because if you actually break down that problem, first I have to make the cargo fit in the vehicle and fit in a way that it keeps the cargo stable and doesn't damage it while it's in there. Next, I have to think about, okay, I do this subcutaneously. How do I make it so that all of that vehicle doesn't just deliver to the cells in the subcutaneous space, so like under my skin where I inject the medicine, and actually then doesn't go anywhere? So that's your first hurdle. Then I need to get it to the blood-brain barrier, somehow get it across that blood-brain barrier without again, like just sticking in all the cells of the blood-brain barrier when I get there. And once it gets across to that other side of the blood-brain barrier, I need to figure out a way to get it to neurons specifically. And so we, for a long time as an industry and an academic area, have been working on these challenges, right? And I think the great thing, and the reason why you're seeing sort of this massive uptick in genetic medicine broadly across our industry and across academia, is because we're getting closer to the solutions of some of those problems. Yeah. We shared at a recent conference some data that we have here at Lilly on our blood-brain barrier shuttle technology, where now we have an approach where we can conjugate something to our siRNA and have it ferry that across the blood-brain barrier and knock down genes in the brain of a mouse. 10 years ago, that wasn't possible. A couple of years ago, that wasn't possible. But now we're crossing these frontiers, which makes some of us, me especially, really confident that we're getting closer and closer to taking these to patients.
It's an exciting time, and it's fascinating. It must be fascinating being on the forefront of doing all this. One other sort of career stop for myself, maybe about 15 or so years ago, was with a recombinant protein production company, and sort of the thesis at the time was, wow, we have all these blockbuster small molecules today. In 20 years, it's all going to be genetic medicines and antibodies and so forth. We need more production capacity. And, here we are. It's 2022. I look at the top 20 therapeutics out there. It's all antibodies and so forth. What are some of the things that you need to think about? And I don't know, we're talking about 2030 or 2030, whatever, as far as production scale up of some of these very unique moieties, let's say.
Yeah, that's a really pertinent, relevant question, right? I think these modalities to date have, like I mentioned earlier, mostly been applied to rare diseases where, best case scenario, you're talking about patients in the tens of thousands in terms of economies of scale of making things cost effectively. Like it's tough when you have very small populations, right? Because you're doing sort of bespoke manufacturing type methods to make the material to deliver to the patients. And anytime you do things in sort of more of a one-off type way, it gets more and more costly, because those platform type innovations on the manufacturing side are how we bring the cost down, right? We do a lot of things in a very similar way, and then the cost comes down over time. If you have to do unique things a lot, it can be challenging to get those economies of scale. I think the good thing that we're going to see over the next few years, and I think COVID actually accelerated this a lot, right?
Yep.
You can imagine it globally massively increased the market for RNA-based medicines. And so it massively also increased the production capacity for RNA-based medicines. And that for us in, beyond the vaccine field is going to be a big boon as we sort of try and take different kinds of RNA medicines to patients over the next few years. There are still, a lot of innovations that are required though to make this as cost effective as we would like. And oligonucleotides today, if you look at, the field in general, are probably still one of the more expensive things to make that exist in terms of the therapeutic options that are approved for patients today. There's, I think, 6 or 7 approved oligomedicines today. So it's not at a point, if you exclude the COVID vaccines, it's not a point where you have that economy of scale yet. And so I think we want to be one of the companies, though, that are pushing the boundaries of manufacturing too. And so, one of the groups that we have at the Institute is part of our CMC related organization. And the focus there really is on pushing the boundaries of what you can do in terms of cost effectively and scalably manufacturing oligonucleotides. Those people are working on, really cutting edge ideas that take us beyond the solid phase synthesis that people are working on today. And solid phase, just to give people an idea of what I'm talking about there, essentially you have a bead, a polystyrene bead that you then build one by one your oligonucleotide from base number 1 all the way up to base 21. You can imagine that doing this one by one manufacturing is a very time intensive, labor intensive, not very cost effective way of producing things, but it is well trodden and people understand how to do it. And so in the space, that's what we're working with today. There are a lot of people that are working on more interesting approaches, looking into things like fragments and making, if you have a portion of your molecule that stays the same, can you make a large amount of that piece in a different way and then use some reaction to conjugate it to the variable piece of the molecule and cut the cost that way? Or, could you think about something other than solid phase as a way to glue your one by one reaction together and make it more efficient that way? And so for us, we're thinking a lot about how we can be part of pushing the boundaries there too. And the Institute in general, like the big focus that we have there is really about how do Lilly help push the boundaries of science in the oligonucleotide and genetic medicine space. That's going to range from everything to like, how do we push boundaries on the computational side, like we talked about earlier, all the way through to how do we make these medicines more cost effective? I mean, what we want to do as a company is reach as many patients as possible with the medicines that we make, right? That's why I do this. That's why my team do this. We measure ourselves on the number of people that we help. And so I think for us, like making this more efficient is absolutely one of the ways that we can help more people.
And, continuing along that thought process, in the recent press release about the Lilly Institute for Genetic Medicine, it also mentions a very unique incubator model as part of the system. And I thought this was fascinating because I think too often nowadays we hear, pharma has gotten out of early stage research, or, if you're coming in with something you want to partner with big pharma, don't bring it to me unless there's phase 2 data and all this stuff. You're really looking to partner with a lot of the really early startup innovators in this area. Talk a little bit about how this model works.
Yeah, I'd love to. I mean, I'm really fortunate. I've got to sit on the board of a number of companies in this sort of really early space. And, what you learn there is that if we can help with specific resources or specific expertise that you have at a large pharmaceutical company that may be challenging for people to get when you just start your biotech. I have people that I get to work with on a daily basis that have been thinking about, manufacturing and CMC for 20, 30 years, and they have a ton of experience in that space that lets them problem solve really effectively. The idea with the incubator is to bring in companies primarily aligned with the genetic medicine mission of the Institute. So they're going to be people focused in similar areas that we're interested in, that are looking to leverage sort of Lilly expertise to help them accelerate their mission as a company. And I think, the great thing for me and for the Institute is those companies are going to come in with their own perspectives, their own points of view, like diverse ways of solving problems, and just exposing, people at the lunch table to a conversation with a scientist that they might not meet otherwise, right? And say, have you read this paper? Like, that's super valuable to us. It's really this concept of diversity of thought, I think, is really important to us as a company, and bringing in early stage people. There are challenges, obviously. It's always much easier the later you go in the drug development process, the lower the risk is in terms of external innovation. But in genetic medicine, there aren't many companies in the world that have a ton of experience really at this point, right? It's a field that's not very old. So really a lot of the external innovation I'm going to be doing, regardless as to whether or not, it's part of the incubator, is going to be focused on that early stage. The incubator, though, gives this proximity aspect as well. It makes it easier to have conversations with folks at those companies, easier to collaborate, and easier to share ideas of how we can help them. So we've done this before. We have a facility in San Francisco, Lilly Gateway Labs, where we've had some really successful companies come in and I think do some really cool science, and some of that in collaboration with folks at Lilly. I'm really excited to recapitulate that model here in Boston. It's probably going to be even more intense in Boston, just given the number of RNA and DNA-based companies that we have there. It's a super exciting time for us, and I'm really looking forward to see who we can bring into that space.
Awesome. Yeah, and it's just refreshing to see this sort of this hybrid model, and that you have the vision for this. So it's really, really fascinating stuff. Andrew, one thing I always enjoy is that in the beginning I went through sort of your couple hundred or so publications in PubMed, and seeing sort of the different themes that you've been focusing on and focused on earlier in your career, and there's always one that falls out. And for you, it happened to be this paper from, I think about 10 or so years ago. You were working with Rhonda Pinhow. And this paper was entitled Telomerase Reactivation Reversing Tissue Degeneration in Aged Telomerase Deficient Mice. The reason I bring this up, obviously, Lilly, a leader in diabetes, a leader in the neurosciences, we've been doing a lot of episodes on this area of what I term longevity biotechnology, sort of this evolving area of companies that, is not looking to necessarily target Alzheimer's or diabetes, but sort of looking upstream at sort of the processes behind aging, obviously telomeres. I would just love to get your top line for a couple of minutes on your view of sort of the aging biotech space in general. Obviously, there's confidential stuff here that Lilly's doing. You don't talk about it, but just like to get your thoughts, being in a sort of a moonshot organization within Lilly doing this amazing frontier stuff. Thoughts on aging as an interesting target in the future, aging biotechnology, things along this line.
Yeah, it's a really hot topic right now. Right? I've been, for a long time, you pulled out one of the more interesting papers that I think I got to do while I was in Boston. And it really is a topic that when you dig more into it, it's absolutely fascinating, right? Like, we're sort of barely scratching the surface, I think, of how the aging process works. And I follow people like David Sinclair and other folks in the field, and the stuff that they're doing around the Yamanaka factors and NAD and other things like that. For Lilly, one way to look at this, we may not be in aging right now. We may not be in regenerative medicine per se, but you could imagine that if you were forward looking at a company that works on diabetes and works on Alzheimer's disease, and you believe in your medicines, tirzepatide, the molecule that we're about hopefully to bring to patients for type 2 diabetes and potentially obesity, that's going to transform the practice of medicine personally, right. I think, given the amount of weight loss that's possible with those kind of next generation incretin medicines.
Yep.
So say you're transforming weight loss, you're reducing cardiovascular risk. Donanemab is another molecule that we have for Alzheimer's disease that I'm obviously really passionate about too. And if you can start to make an impact in slowing the disease progression or halting the disease progression in Alzheimer's, which is sort of my personal mission, then the next step logically after those two things is that you're going to have an increasingly aging population, because you've removed some of the larger factors in mortality in those populations, right? And so it would make sense logically for companies to, as the next phase, start thinking about how do we now, since we've removed some of those things, tackle the inherent biology of aging, right? And how cells and tissues and systems break down over time, and how can we either prevent that or restore that. The science has come a long way since that telomerase paper originally, and I think that there are a lot of very recent interesting papers, especially in rodents, showing that using various combinations of gene therapies for things like the Yamanaka factors, that you can turn back the biological aging clock in rodents. You can maybe take cells into different states that we didn't think were possible, or we thought if it was possible would lead to potential sort of mitogenicity and cancer risk. And we're seeing that sometimes maybe that's not the case now. So I wouldn't be surprised if the next horizon that you see a huge explosion of in the biotech space and then in the industrial space really is this, how do we tackle aging problem? Yeah. There are people who've been thinking about it for a while. You probably thought about some of the same people. The Buck Institute and people like that have been working on aging for a very long time. But I really do think it's sort of primed to come mainstream over the next few years. And that's going to be a very exciting time for genetic medicine, because really, from my perspective, these are the kinds of tools that you would use if you wanted to modulate the aging process or sort of try and slow the aging process. You'd do it at a transcriptional level, probably.
Yep. Yeah, I was. It's interesting you brought up the Yamanaka factors, and I've been thinking a lot about that, especially with some of the tools that you're developing at the Institute. But then, also folks like George Church, who I interviewed a couple of years ago, who talks about, 20 or some odd genes that he wanted to. So it's going to be an interesting time. It's going to be really fascinating to see how what you come up with at the Institute feeds into sort of these multiplexing is the right term, but these really sort of combinatorial interventions when it comes to complex regeneration and, of course, the aging process. And that's really very exciting. Andrew, early on in our discussion, you mentioned, obviously, you've had an amazing career at Lilly. You've been mentoring folks along the way. Take a couple of minutes just to talk about some of your mentors, if you would. Some of the folks that have been there. Obviously, you worked with Ron DePinho, a bunch of other fascinating people along the way here. But who's been really instrumental in keeping you on this path and keeping you excited about all this stuff? And the science obviously keeps you excited, but some of the people, if you could talk about them too.
Yeah, for sure. I've been really fortunate through my career, and that's kind of why I feel like I need to pay it forward now with the mentoring that I do. I was very fortunate early in my career to work with Terry Flyer in Boston, who was my supervisor when she was at Beth Israel. At the same time, I got exposed to Jeff Flyer when he was the dean of the medical school. We were really lucky that we would have our team meetings in Gordon Hall at Harvard, which was sort of a very unique experience to go into sort of this really old building and have lab meetings to discuss data. When I moved to Lilly, I was very fortunate that Tim Garnett, who was our longtime chief medical officer, was one of the people who sort of took me under his wing as a fellow British person at the company and really helped me grow in my career here at Lilly. Ruth Jimeno as well, our vice president for diabetes, was my boss for 8 years, is still a longtime mentor of mine and someone who I learn a lot from. So, what I've been really fortunate to be exposed to at the company is just a really diverse set of people with a lot of different ideas and different backgrounds. That's, Lilly is a fantastic place because we have people from all different backgrounds and from all over the world. And this is, I guess, one of the pluses of a large pharmaceutical company. Just the sheer number of different perspectives is fantastic. We're trying to build on that in Boston. One of my personal passions is sort of diversity, equity, and inclusion. And I think with the hiring at the center, you're going to sort of see that reflected, right? Bringing together a lot of really unique, different skill sets from different people with different backgrounds. And I think it's really that diversity that has powered the kind of innovation we've been able to achieve over the last couple of years. And it's going to be a huge growth for us too, right? We're going to go from 120 people now to more than 250 people in a few years' time at the center. And that's a huge opportunity to bring in a lot of really interesting science and a lot of really interesting people to the company.
Mm-hmm. Mm-hmm. Yeah, it's going to be exciting times. Andrew, final question. As we've been talking, you've been sitting beneath an image of a T-Rex. You mentioned your interest in paleontology at the beginning, and it's funny. I had Dr. Dana Rashid on the show about a year ago, who's making the chicken dinosaurs out at University of Montana. All that being said, If tomorrow you were to come into the Lilly Institute of Genetic Medicine and find out, voila, you've cured all degenerative diseases, you've dealt with aging and regeneration, all this stuff. Okay, you've taken care of all that. What is Dr. Andrew Adams doing next? Are you getting back into paleontology? What else excites you when you go home at night and don't want to think about drugs?
Yeah, my plan. It's a really interesting question, and actually, it's kind of funny because I know Dr. Rashid's work, know a lot about the chicken dinosaur stuff. It's actually really cool that they have all these latent genes that you can turn back on again. Yep. My life after Lilly, I would imagine it's going to incorporate some teaching probably somewhere. I'm really passionate about teaching, and so hopefully we'll have an opportunity to do that somewhere. The other piece, I hope I get to work outside in the sunshine, like digging up some dinosaur bones somewhere, and get to it after 20, 30 year diversion and go back to doing that again maybe. But I'm just really lucky, today, I think like a lot of us, my life has been touched by Alzheimer's disease and some of the things that my department here work on at Lilly. And so for me, the work's not done until we make a significant impact in some of those areas. And that's my goal. And I know a lot of the people in my department's goal is to make a big impact on things like AD and Parkinson's disease. So I've got a lot of work to do before I go anywhere else yet.
Well, you'll definitely get there, and I'm a big fan and rooting you on. So, this is such an exciting story, Andrew. As I said, really wishing you the best with all of it. Obviously, your team, the team as it grows. For everybody that is going to be listening to this particular episode of the show on our various podcast networks or watching on the YouTube channel, you've been listening to Dr. Andrew Adams, Vice President, Neurodegeneration Research, co-director of Eli Lilly's new Lilly Institute of Genetic Medicine, doing really amazing things to drug the undruggable, open up these new areas of clinical investigation, and ultimately impact patients and people's lives. Andrew, I want to thank you again for taking the time out of your schedule to come talk to us for a little while. Obviously, thank you for everything you're doing there at the Institute. And we say on this show, thank you for helping to create a better tomorrow through everything you're doing. It's a really wonderful story.
Awesome. Thanks, Ira. I really appreciate it.
Be well.